Okay, everyone. So hopefully you can see my screen. Rosie, can you see it okay?
Yeah, thank
Yep,
you.
okay. So, yes, as Rosie says, I've worked in clinical trials for more time than I would like to talk about, and I have put together a presentation today to talk about some of those terms that are used all the time by people like me, but aren't necessarily day-to-day conversations. Now, I realise that perhaps I use this all the time and I don't actually know that they are rules. Because of that, I thought it would be helpful to collect sort of there might be terms that I don't cover because either they're eye related and I haven't thought about them, or they're terms that I use all the time and I forget that not everybody else knows what I'm talking about. So what I did is I started a Mentimeter. And if you can either go to menti.com and put the code in that's on the 22488908 and use the code. And then if you go onto that website at the same time, then you can type in any words that you'd like me to explain that I might not have forgotten. Alternatively, you could put it in the chat as well, and we'll go back to the chat at the end of the presentation to see if there's anything in there. I do a quiz at the end, but we'll see how many people sign up and see that works all right. So that's good. If anyone's got any trouble, let me know. Okay. So I would have a hypothetical clinical trial participant, and we're going to call her Sarah, and she's going to take part in a clinical trial. And I'm going to walk through the process that she's going to go through, and we'll talk about some of those terms that she'll come across in her journey. So first of all, there's the research team. So she'll be probably invited to a hospital or to a doctor's office, and she'd meet the research team, and that's made up of various different. First of all, the investigator is the doctor who leads the study within that hospital, and usually the clinician that's responsible for Sarah, for your care or for Sarah's care for the person taking part. The term investigator is it has sort of in ICH GCP, which is the rules that we abide by. It's an investigator has responsibilities that they have to take care of, and if you go back into the YouTube channel for starts, I did a talk a while ago about trials, and it runs through all of the investigators' responsibilities. You might see terms such as principal investigator or co-investigator or sub-investigator. And principal is the one definitely in charge at that hospital, but he may work with doctors and other allied healthcare professionals, and they may be double co-investigators. And then not at the hospital, but otherwise, you might see a sponsor or a contract research organisation referred to. The sponsor is the person who, the organisation that's running and funding the trial. So all the money comes from the sponsor, and they are also the group that's legally responsible overall for the study. So sponsors tend to be either pharmaceutical companies or biopharmas, but sometimes they can be universities or charities or hospitals themselves can be a sponsor, but they're the ones that the buck stops at, who are ultimately responsible. And lastly, a CRO or a contract research organisation. Sometimes these sponsors don't have all the resources that they need in order to run a study. And if that's the case, they might hire some of those responsibilities out to another organisation, and that's what's called a CRO. That's who I work for. Basically, we're parts of the study. So they might do data management, or they might look after the logistics of the drugs, so getting the drugs to where they want to be, or they might who checks the data is what it should be in this, and that's called monitoring. So that's the contract research organisation, who may be responsible for a little bit of the study. Sometimes they're responsible for the whole thing as the sponsor. It's doing the organisation. Okay, so unless there's any questions about that slide, we'll go to the next one. So Sarah hears that she might be eligible for a few different study types. So she receives an invitation to go to the hospital, and she goes, and the research staff at the hospital tell her about the different types of trials that she could take part in. Now, each of the clinical trials that she could take part in, they have a title and they use certain terms. And those terms mean things to the people who work in the research, but not necessarily other people. So there are different study types. The title might have the word interventional in it, and interventional means that there's some sort of intervention. So usually that means that Sarah would receive some sort of treatment. So either a medicine or an implant, and that's the intervention, and that makes that trial interventional. Alternatively, there might be an observational trial, and that's where there's no intervention, and taking part of the clinical trial won't mean that she receives a particular treatment that's different to what she would normally have. And what happens in an observational trial is that researchers just track Sarah's health over a period of time, and sometimes they even go and look and see what's happened in the past. So there's no treatment given, but there may be a blood test or some eye test or something else that the researchers use to measure Sarah's health, but there's no particular medication. One particular kind of observational study is called a natural history study. And I brought this one up because I know that it's been a term that's been used by some of the sponsors and the biotechs that have spoken in other webinars for this group. And a natural history study is a long-term study that tracks how a condition progresses without treatment. So very similar to an observational, it's just a type of observational. And it's tended, for Stargardt's, it will tend to be done by sponsors that are running a gene therapy trial. For gene therapy trials, and we'll talk about arms and stuff later, but usually in a gene therapy trial, there's just one group of people. And if you're in the trial, you get given the gene therapy. And what that means is that there's nothing to compare how well those people are doing compared. There's no data to compare them to. You just know how well they're doing, but you can't say whether it's better or worse than they would have done otherwise. So if this is the case, what sponsors often do is they run a natural history study that runs just before or sometimes at the same time as their interventional study, and they use the data of their natural history study to compare what the outcomes are for those patients. So in a Stargardt study, they would probably see the patients that were in their interventional study and had the gene therapy, and they would look about how their vision was and how they measure their vision, and they would compare it to the people in the natural history study and see whether it is better, the same, or worse. And then another kind of study is a registry, and that's more of like a database. So Stargardt's Connected has its own registry where we collect information in. And a registry is really a long-term database where anyone can go in and put in their information, and it tracks people with a certain condition or after a certain kind of treatment. It's not as robust. So in the natural history study, you would go to the doctor just to have your eye test for the study, whereas in a registry, it probably wouldn't have that level of detail. And if it did have that level of detail, it would be an eye test that you were going to have anyway. And then the last word I've got on this is protocol. So the protocol is like the recipe book that every investigator has to follow. So quite often clinical trials will be run at lots of different sites, but all those sites will be having the same protocol. And it's the detailed rule book guiding all the aspects of the trial to make sure that everybody is doing the clinical trial in exactly the same way, is recording the data they receive from that in exactly the same way, and that everyone is following the rules around safety. So the patients are all safe as well. So another thing that we quite often see in the title of a clinical trial is the phase of the study. And typically you see phase 1, phase 2, phase 3, and phase 4. And these are different studies that run sort of one after the other. So normally you start with a phase 1, you have a phase 2, then a phase 3, and then a phase 4. Although there can be some that run in parallel, it's not necessarily in that order, but typically it's in that order. Now, phase 1 study is around safety and dosage. So if a sponsor has a medication that they want to try for the first time and they haven't used it in humans ever before, but they have some information from animal models, there's always going to have to be the first person who takes that at some point. And the phase 1 is the first in human where the first people take it. So the focus on here is it's a very small number of people. Typically, what we see is a 3. So you try 3 patients on a particular dose. And if everybody's all right in that dose, then you move up a dose. And that's kind of the way that you do it. So a phase 1 may only have like 9 people in it. And its focus is on safety to make sure that nothing unexpected or untoward happens. And also to try and figure out what the best dose is before you treat many more patients. Sometimes this is done in healthy volunteers. It's only really done in patients where there is an expectation that it will be highly toxic, or perhaps it's a really invasive way of getting that treatment, or if it's a permanent change. If it's going to make a permanent change, then it would be done in patients. But normally it's done in healthy volunteers, and they have these little centres where students and things sign up and go and live there for a week and have their blood tests, and they all have to collect all their wee and give them all in and all this kind of stuff. So it's kind of, these special centres where these studies get run when it's in healthy volunteers. And then those students get paid for it and they keep the money. So a phase 2 study is sort of the first kind of time when you say, is this drug going to be effective? So by phase 1, you know what dose you want to go with, you know how safe the drug is and what the likely side effects are going to be, and that they're not going to be too terrible. Then the phase 2 is generally when you first go into a patient, and this evaluates how well the treatment works. And it does it in a fairly small number of patients. So you might be looking, I don't know, about 50, it depends on the indication, but it's got a larger group than the phase 1, but still not a huge group. And it really it's sometimes called a proof of concept study, where you give it to the patients and you see, does it have the effect that you wanted it to have? And if you pass your phase 2 and you see the effect that you were looking for, then you can go on to a phase 3. And for most drugs in the market that start on this journey, most of them fail at the phase 2 point of time. This is the sort of one of the key points in seeing whether you're going to move forward with this trial, with this drug or not. So phase 3 is where you confirm that it works. So you give it to a much wider population, and you compare it to standard of care, and then you see if it works or not. So I don't know if you remember, during COVID, there was lots of talks about the vaccination trials. In those trials, they had like 50,000 people. Half of the people got the vaccine and half of them got a different vaccine that wasn't for COVID. And then they followed those people and they said, out of those 2 groups, which ones caught COVID most often and were iller with their COVID. And that's how we were able to give the vaccine to everyone, because the group that was on the vaccination didn't have as much disease and it wasn't as severe as the ones that were not on the medication. And that's the phase 3, where you use a big trial, where if there's a rare side effect, you're more likely to pick it up then because you've got more patients, and it really confirms whether it works and makes sure there aren't any side effects that only a small number of people have. And then phase 4 is called a post-approval study. So after the phase 3, the drug company will go to the regulators and say, can we sell this to people without it being in a trial? And the regulators, some say, yes, you can, but there are some things that we want you to monitor even though you're selling it to it. So sometimes that tracks long-term safety. So in gene therapy studies, they have to track long-term safety for much longer than you would for other studies. Or sometimes it looks into a different population. So maybe you have done your trial in adults only, but there's a big population of children that have the same indication. And so a post-approval study may be in children. So those are a couple of examples of phase 4 studies. So according to ICH-GCP, and I think we cover that in a bit later, which is the rule book that you run clinical trials by, Sarah must give her permission to take part, and she must understand what taking part means. So Sarah will be given a patient information sheet, or a PIS, and basically that's a leaflet that tells you everything about the drug that you'll be taking, and everything about the risks of that, but also how often you'll have to go and see the doctor and what tests you'll have to have. She will then sign an informed consent form, or an ICF, and basically that is a document that says, yes, I've read the patient information sheet, I'm happy with the information that I've seen and I understand it, and I've had a chance to talk to someone about it, and I give permission to be part of this clinical trial. So you should never do anything to do with the clinical trial before you signed your informed consent form. And ICH-GCP it stands for International Committee of Harmonization. Good clinical practice is what it stands for, but we only ever use a little one, and it's a global rule book. So it has been agreed across the world, and it is the rule book that says who's responsible for what and what things you have to do in order to make sure that we're protecting people that are taking part in clinical trials, and to make sure that the trials are of good quality, because there's no point putting people through the process of going on a clinical trial unless what you do is good quality. So Sarah may have to take a medication as part of the trial. So an investigational medicinal product, or an IMP, is the treatment that's being tested in the clinical trial. So it's the study drug, or perhaps the study therapy is called the IMP. You can have small molecules and you can have gene therapy. So a small molecule is a traditional medicine that you would normally be used to taking. So it's normally a pill, or maybe a syrup, or perhaps even an injection, but it's a chemical that you're taking that has a certain effect. Then you have larger molecules, and I've got gene therapy here, which is something that uses genetic material to fix, replace, or modify genes. And gene therapy is generally sort of a permanent. So you have it once and then it permanently changes everything. And there's actually a class that's in the middle of gene therapy and small molecule, which is called biologics. And they are larger sort of protein, sort of biologically made things. So if you think about insulin for people with diabetes, and they're in between that. So they last longer than a normal medication, but they're not permanent. And you normally can't take them as pills. You have to have an injection or an implant or something like that. So then the size of the molecule that you'll be taking as part of that medication changes how everything happens during the trial as well. There's a concomitant medication. So the doctors that Sarah sees will ask her what other medications she's taken while she's been on the study. And they will write all that down, because that's all really useful information. And they would include sort of things like vitamins that you might take all the time, or eye drops. And in the protocol of that the doctors are following, it will have a list of allowed concomitant medications, or it may have a list of things that you can't take at the same time. So if the study drug works in a particular way, but there's another medicine that works in a similar way, the doctors won't want you on both at the same time, and that will be in the protocol. But the doctors will collect all the information about all the medications you take, and that will be concomitant medication. And lastly, under the medication bit, I've got placebo. So a placebo is a lookalike treatment with no active ingredients, and it's used for comparison. And the reason that this is done is because they did a whole load of studies, and just by going to the doctor and having attention on you and having lots of different tests on you, that by itself can just make you feel better because you feel like you're being cared for. And that's called the placebo effect. So if you didn't have something to compare against, you wouldn't know that the reason why people are getting better is nothing to do with the medication at all. It's to do with the rest of the care that's going with it. And that's why in these protocols, they have placebo arms. So you have two groups that are being treated exactly the same. And just one of them is getting the active ingredient and one of them is taking a placebo, which looks exactly the same, but it doesn't have the active medication in it. So they will ask Sarah, if Sarah decides and she's signed an informed consent, then the doctors at the research centre will look at her eligibility. And each protocol has a list of eligibility criteria, and that says the things that you must meet in order to be allowed to be included in the trial. So they're called inclusion criteria. So you must be this if you want to take part in the trial. And there's also exclusion criteria. If you're one of these things, then you can't take part in the trial. So good examples for an inclusion criteria was you would have to be a certain age, you would have to have a certain diagnosis, and you would maybe your medication that you're taking on looks a certain way at a point in time. Exclusion criteria are quite often people that have a secondary illness that might confuse the results. Sometimes they're excluded, or if they're taking similar medications like we just talked about. So if you don't fulfill the eligibility criteria, you won't be able to take part in the trial. And the doctors that you see, that's beyond their control. So even if they put you in, that would be something that they're not supposed to do. And the sponsor or the CRO would see that they put you in, even though you weren't eligible, and they would remove you from the trial, and they wouldn't use your data, and they wouldn't provide you with any drug anymore. So it's not completely in control of the doctors that you see. And that's there really for safety as well. So if Sarah fulfills all the eligibility criteria and she's signed to take part, she'll then go through screening and safety. And that includes health checks to make sure the participants are as healthy as you need to be in the trial, and that they definitely meet those inclusion-exclusion criteria. So sometimes you might have a blood test and it shows that your liver is a little bit different to what it should be, and that might mean that you no longer fit the eligibility criteria. So there's a process where we go through screening and safety and we see what Sarah's health's like now. And then once we've confirmed that all the eligibility criteria are met, then we do a baseline. And these are the measurements that are taken just before she starts studying medication. And these measurements will be used in order to compare to after treatment. So your clinical trial may be for 1 year, and Sarah might take a medication for a whole year. So she'll have baseline measurements, and that will be her beginning. And then she'll have the same measurements again after that year. And those are the two things that will be compared in order to say whether the medication has made a difference or not. So before her baseline, Sarah will be randomized. And this is a process. It's like throwing a dice and deciding whether you're going to be in the active treatment group arm or in the placebo arm. So which arm you're going to be is completely luck. And again, out of the control of the doctors that you see. And the name and title of the protocol will give you a clue about how many arms are in, and this information will also be in the informed consent form. A single arm study is a study where everyone gets the same treatment. So if you remember, I talked about a gene therapy earlier, where everybody got the gene therapy. That's a single arm study. A double arm study is where there are two different groups of people and they're getting different treatments. So an example would be a study where you have one group taking active and another group taking placebo. And a multiple arm study might be if you're having more than two different treatments being compared or different dose levels. Again, it's random which one you go in, but there may be more than two, and that's called a multiple arm study. And the randomization process is how we decide which group you're going to be in. So it used to be in the olden days that every investigator had a group of envelopes, and you picked the next envelope and you looked in the envelope and it told you what study drug you were going on. Now it's all just done by a computer. So blinding or masking is the process that goes on so that you don't know which treatment you're on. So if you were on a placebo study and you knew that you were on the placebo, you might say, well, I don't want to be on it anymore, and you might leave the study. Also, it might impact how you're feeling. So we do this process called masking, which means that Sarah, as the participant, doesn't know which treatment she's getting. She just knows she's getting a treatment. And different types of study have different types of blinding or masking. A single blinded study means it's only the participant that doesn't know. The doctor that she speaks to knows what she's on, and all the people that look at data, everybody knows, but Sarah, the participant, doesn't. That's really unusual. I haven't really seen many of those. Most studies are a double blind, and that means that Sarah doesn't know, her doctor doesn't know, the people in the offices that are looking at her data, nobody knows what kind of treatment she's on. And what that means is that when the doctor assesses how well you are, he's doing that without any bias of whether he thinks you should be better or not. And the same for all the other people that look at your data. But there is always a way of finding out what you are on. So if you were to go to an accident emergency and something had happened to you, and those doctors really needed to know what you were on in order to be able to treat you properly, then there's usually like a phone line that you can ring up, and then you have to say why it's an emergency situation. And in those cases, if you need to, you can find out which drug you're on. But we try and avoid that whenever possible, because it means that you can't use your data anymore in the trial because it becomes biased. An open label study is where everyone knows what treatment you're on and there's no secrecy about it at all. So there's and a control group or a control arm of that group that is receiving the placebo. And unblinding is when you ring up and you say this is what your treatment you're on, or if no one's been unblinded throughout the whole of the study, then at the end they sort of unblind everyone when they've already finished. Everyone gets unblinded at the same time, and then the statisticians know who to put in what group, and then they're able to compare them. And usually you should find out after the trial, when it's been unblinded, you should find out what treatment you were on, and that might impact how you're treated in the future as well. Sometimes you're allowed to take the active drug if you've been on placebo the whole time, for example. Right, I've been talking too long now, I've just realised. We're nearly done, I promise. So treatment and safety, an adverse event is any change in health, even if it's a small thing like a headache, and even if it's not related to the medicine that you're taking. A serious adverse event is an adverse event that's really serious. So a major life event, so a hospitalization, something that disables you, or something that's life-threatening. And whenever there's a serious adverse event, there has to be an investigation as to what's happening and whether it impacts the other people on the trial. An adverse reaction is a side effect that we know is linked to the actual study treatment. So if you have an adverse event and they investigate and they say it's to do with the treatment, then it becomes an adverse reaction. And endpoint measures, those are the tests that you have. So we've talked about comparing the people that have had the drug with the people on placebo, and they use endpoint measures to compare. So in Stargardt, that's probably the eye test that you have. So there'll be a number that comes out of your eye test, and that's what they'll compare the two groups from. So this is my last slide. So regulatory and ethics. An institutional review board is normally an American phrase, but it's the same as an ethics committee. And they are a group of people who meet and decide. So they look at the protocol, and nobody to do with the trial is included in that group of people. And they look at the protocol and they say, the doctor at this hospital can do it, and this protocol is all right to do. So they make sure that nothing unethical is happening and that it's being run by people with the right amount of resources. Every sort of hospital has an ethics committee that's sort of linked to it. or sometimes there's like a central one in a country, but there's always some sort of ethics review according to ICH-GCP. The Medicines and Healthcare Products Regulatory Agency, or the MHRA, is something that you may have heard of when talking about new medicines. That's the UK regulator, so the government body that looks at, it approves, it allows clinical trials to go ahead in the whole of the UK, but also is the one that says when the data comes in from the clinical trial, that's the group of people that says, yes, you can sell this in the UK, or no, you can't sell in the UK, depending on that data. And it's different from the National Institute for Health and Care and Excellence, which is NICE. So the MHRA firstly says, yes, you can sell this in the UK. It then gets referred to NICE. And this is the group that says whether the NHS will pay for or use this treatment. So the Medicines, Health and Regulatory Authority are looking at, is this safe and does it work? Whereas the NICE people, they're looking at, is it worth the money for the country to spend on this? Or is it too expensive and it's not worth the outcomes that are? So something might work and you may be able to use it privately within the UK, but it may not meet the thresholds for NICE, which means that you can't get it on the NHS. You have the FDA, which is the Food and Drug Administrator. They are the American version of the MHRA. There's also a different one for Europe, and every different country has a different body, but those are the ones that you're most likely to hear about. And a marketing authorization is that official license that you get from the MHRA that says you're allowed to market a particular product, and it's at that point that a doctor can prescribe it. And I feel like that's a lot, Bhavna and Rosie, but those are all my slides, and I realised that we've gone quite long, so I don't know if we want to leave it there rather than doing the Mentimeter.
I think we're okay for a bit of time